Can you see the screen?
Yes, Doctor.
Yes, doctor.
Okay, so Bismillahirrahmanirrahim. So the topic for the today's session is systematic review and meta-analysis. So I think some of the students, I think group A and B have already attended today's session. They must have had the practical part about the systematic review and meta-analysis. So this is just introductory session because after this session you are supposed to do the practical session. So group A and B have already done both on the male and female section. So we should have some basic idea that what is the meaning of systematic review and meta-analysis. So this is the purpose of the session, that you understand the concept of these two terms. So the main learning outcome is that you recognize the different types of studies which are used in clinical research. So I think already in the introductory session to the EBM or the evidence-based medicine, you know what are the different types of study designs, and this is also important in terms of your PEP or the professional excellence pathway, because you have to do review of a study. So you should have some basic idea about the different types of study designs which we are using in the clinical research. Yeah, so as you can see here, this is called as the pyramid of the EBM or the evidence-based medicine. As you can see in this pyramid, there are different types of study designs, starting from the test tube researches, or which are also called as the in vitro studies. Then we have the animal research, we have ideas, editorials, opinions. Then we have the case reports, case series, the two types of the analytical studies, that is the case control studies and the cohort studies. Then further up on the pyramid, we have the RCTs or the clinical trials, which are called as the randomized controlled double blind studies. So the other name for RCT or the randomized clinical trial is the same RCT, or you can call it as the double blind studies. And on the top of the EBM pyramid, you can see the top position is occupied by systematic reviews and meta-analysis. So can anybody tell me that the higher up the pyramid will we go, which studies are good for us in research? The higher up the pyramid or the top of this pyramid, this study is good or the bottom is good?
Top, okay, absolutely.
Top. Yes. Yeah. Good. So, so as we all know that the higher up the pyramid, as we go from the bottom to the top, the quality of the study keeps on improving. And the studies at the apex, that is the systematic reviews and the meta-analysis, they will give us the best evidence. The best evidence or the best clinical evidence which we get by randomized clinical trials and by systematic reviews and meta-analysis. So the top position of this pyramid is occupied by RCTs, systematic reviews, and meta-analysis. So if anybody asks you in the exam or maybe in the Viva that which studies will provide you the best evidence, so your answer should be systematic reviews, meta-analysis, and RCTs. Okay, so these they provide us the best evidence in the clinical research. Whereas the studies which are at the bottom, like the animal researches, the ideas, editorials, opinions, though they also provide some evidence, but it is a poor evidence. The evidence quality is not that much good. We can't generalize the editorials or opinions. So the quality of evidence is at a lower side in them. So therefore, this pyramid is called as the pyramid of the EBM or the evidence-based medicine. The higher up the pyramid we go, the better is the study, or these studies at the top will provide us the best evidence. So this is just you should have. I'm not going in detail of these studies because I think it has already been covered in the earlier sessions. You should know the differences between the randomized clinical trials, what are the cohort studies, case control, case series, case reports. So I don't want to repeat them. Today we will focus only on the systematic reviews and meta-analysis. So just to tell you in very briefly, what is the meaning of systematic review? So basically, systematic review is a protocol. It is a protocol driven comprehensive review. So it is a comprehensive review and synthesis of data. We are basically doing analysis of different types of studies, and we are combining their data. We are combining their data. We are doing the synthesis of data, focusing on a specific topic. So the idea behind systematic review is that we are studying the review of any one topic. The topic should be specific. It's not that I'm studying different types of research questions or different types of study titles. So I am focusing on a specific topic or any type of key question, and this type of systematic review is typically performed by experienced methodologists, or it can be researchers or statisticians in their domain. And the first step which we conduct in a systematic review is to formulate a specific question. So first you have to formulate a key question or a specific question about which we are going to gather information. So what is systematic review? It is basically overview of a particular clinical research question. So whenever there is, by clinical research question, I mean that what is the study title, the title of your study, or the research question, what you are going to study or analyze or evaluate. So that is the clinical research question. So you are basically doing the overview, or you are studying about that particular question in detail. You are trying to study all possible aspects about that particular clinical research question. It is almost similar to meta-analysis, but the difference between systematic review and meta-analysis is that the systematic review, if it is done using the statistical methods, then it is called as a meta-analysis. So meta-analysis, it uses a statistical method to summarize the studies, whereas if we are doing only systematic review, it may involve the meta-analysis or may not also. So if we are doing the analysis of the different types of studies, we are summarizing the different types of studies by using a statistical method, then it is called as a meta-analysis. So what, uh, as I told you in the, as you can see, the pyramid of the evidence-based medicine, we are conducting different types of RCTs or randomized clinical trials. Suppose there are 5 different types of RCTs done on a specific research question. So I am going to analyze the findings of all these 5 randomized clinical trials, and I am trying to review their matter, and that is called as a systematic review. And if I am going to analyze these 5 different types of RCTs by using the statistical method, then it is called a meta-analysis. So simply, a systematic review along with the statistical techniques or methods, it becomes meta-analysis. So this is just a basic difference between the systematic review and meta-analysis. So often, as you can see, often the meta-analysis is included in a systematic review, because often in the systematic review we are using the statistical methods. So then the systematic review will become meta-analysis if we use the statistical method. So I think it should be clear to you now. And in the meta-analysis, because we are using a statistical method, it will produce a new single quantitative result, because we are using the statistical methods to summarize the findings of all the different types of data which we are going to analyze, and we get a single value or a single quantitative result. You will understand once we come to the examples. So basically, in the meta-analysis and the systematic reviews, we are getting the pooled treatment result, because we are doing analysis of different types of studies, different types of randomized clinical trials, and we are pooling their results. We are summarizing their result, we are combining their results, and we are looking for the overall effect estimate. What is the overall effect of these different types of values? So both the systematic reviews and meta-analysis, they are more or less interrelated. Only thing in the meta-analysis, meta-analysis will always involve the statistical methods, whereas systematic reviews may involve and may not involve also. Both things can happen. Yeah,
Doctor,
yeah,
the, in the systemic reviews, you
yes,
said that it's overview of clinical research. So if
yes.
I use the same method, systematic review, but I overview of another systematic reviews. You understand me? Like I did, uh, because in our poster we didn't look for clinical research. No, we look for several systematic reviews, and we gather
Yeah, that,
information,
that
and
is
then
yeah, because the poster which you are doing in PEP is not exactly a systematic review. It is more or less a narrative type of review, or you can say the review of the literature. Because I will tell you further in the slides that what is the difference between the systematic review and narrative review. Because the students usually in the PEP sessions they are not exactly doing the systematic review. The review which you people are doing in the PEP, this is called as the review of literature or the narrative, because you are just looking for the material and you are combining the results. Because in the systematic review always there should be a specific methodology, a specific study designs, and some inclusion exclusion criteria, critical approach. So many factors are involved to conduct a systematic review. So like in the PEP, usually the students are doing the narrative type of review. I will further tell you in the further slides, it will become more clear. Yeah, so as you can see in this slide, how we are conducting the systematic review and meta-analysis. Suppose, as I told you, that in the systematic review, we are studying different types of randomized clinical trials. So suppose uh, there is a research title like I want to know the efficacy of corticosteroids in pregnancy. Suppose this is my research question or the study title, the efficacy of corticosteroids, or you can say the role of corticosteroids in the pregnant mother or in pregnancy. So suppose I searched on the internet, on the web, on the PubMed, and I found four different types of randomized clinical trials who were studying the effect of corticosteroids on the pregnant mother. So I found four different types of studies. Now what I will do, because depending on my research question, I have to select the suitable type of study, because on the internet you will find thousands of studies. So the question arises how you will select the best study. So you will select those studies which meet your study criteria. That depending on your research question, we have to select those studies which are more or less similar to your study design. So like suppose we selected four different types of randomized clinical trials and we combine their results. We combine the results of study 1, study 2, study 3, study 4. And if we combine only the results and we study their patterns, their effect, their limitations, then it is called as a systematic review. But if you are combining their results using some statistical method, if you are using the statistical analysis, then this will not be called a systematic review. This will be called as the meta-analysis. So this is just a basic small difference between systematic review. We are reviewing both. In meta-analysis also we are reviewing the different studies, but in the meta-analysis we are doing the, or you can say we are reviewing the studies by using the statistical methods or statistical analysis. Then it is called as a meta-analysis. And if we are simply just summarizing the results, we are combining the results and we are commenting on the outcome or the pooled effect, then it is called as a systematic review. So this is just the basic difference between the systematic review and meta-analysis. So systematic review is an overview of a specific topic. The topic should be specific or the research question should be defined that attempts to cover all of the high quality research evidence that is relevant to that particular issue. So the same thing which I was mentioning, that in your topic should be specific. Then depending on your research question, you have to identify and you have to select all the high quality evidence which is available, or the evidence may be in the form of the different types of clinical trials or the randomized clinical studies that is relevant to the issue or the topic which you want to study. So this is the systematic review. So it is a formal, systematic and structured approach So it is a structured approach to review all the relevant literature. So you are going to review all the relevant literature on the topic. You have to gather the information, you have to evaluate it, and ultimately present the evidence. So some systematic reviews are also the meta-analysis. Once they are using the statistical method, then that systematic review will be now called as a meta-analysis. So as you can see in this figure, like then in the systematic review process like this, there are hundreds of randomized clinical trials in an unstructured way. So what systematic review process is doing? It is arranging with them in the systematic format. So therefore, the name is systematic because we are organizing it. These are haphazard. This is, as you can see, all the studies are arranged in a haphazard manner. We don't know. So we are selecting only those studies which are meeting our study criteria, and we are organizing them in a proper structured. Therefore, it is mentioned it is a formal, systematic, and structured approach. As you can see, it is systematic, structured, and we are gathering and combining the results and then commenting on the findings. Now, in this case, if we are using the statistical method, then the same thing will be called as a meta-analysis. Yeah, so as it's mentioned here, that in the case of normal reviews or narrative reviews like you are doing in your PEP2 narrative review, so we are just checking that are the publications that describe and discuss the state of a specific topic or theme from a theoretical and contextual point of view. So we are just describing the findings theoretically. We are not using any scientific evidence behind it. So there is why there the difference between narrative review and systematic review is the same, which I told you that the narrative reviews, they don't usually use systematic methods, because if we are using the systematic methods, then it will be called as a systematic review. So they are not using the systematic methods to identify, to assess, and to synthesize the information which we are trying to find out. And normally there are no methods section describing how the review was conducted. Like you are making the you're doing any study in your PEP2. So the students are not supposed to describe the methodology, like any whether they have used any type of study design or any type of particularly the inclusion or exclusion criteria on the topic. They are just describing the different types of review methods which were given by the different authors. So it is more or less called as a narrative type of review and not the systematic review. So to start with, like as students, we start with the narrative review, then gradually as we become more experts in our field or good clinical researchers, we keep on increasing our research quality till we start doing the systematic review. And uh, always it will give us the good evidence, because as I told you, the systematic reviews and meta-analysis, they are at the top of the evidence pyramid. So gradually it will keep on improving. So systematic reviews is analyzing the different types of studies, whether they are the human studies or the animal studies, and ultimately it is combining the different findings into a structured format, and for which we can try to find the evidence in the conclusion. So the meta-analysis, as I told you, that in the meta-analysis always there will be a statistical technique. Always there will be a statistical technique for combining data or pooling data from several studies. So the findings or the results or the data from different types of studies or the clinical trials, the RCTs, they are combined. They are combined together and till we get a common quantitative result. So it's a quantitative approach for systematically combining results of previous research. So the study has already been done. Suppose 10 different authors have done 10 different types of studies and there are 10 different results. So what we are doing, we are combining all these 10 results into, and we are analyzing it till we get a common overall estimate or a common result in the conclusion. So it's a quantitative approach for systematically combining the results of previous research to arrive at conclusions about the body of the research. So in the meta-analysis, since we are getting a data it is involving the numbers, so it is called as a quantitative method. It's not a qualitative methodology, it's a quantitative method. The methodology is systematic, like in the systematic review, we are combining the data. So we are combining and putting together all the information. Then the study has already been done. We are not doing anything new. We are not doing a new study. We are just analyzing statistically the studies which were already done before. So we are analyzing the previous research, and in the conclusion, we are getting a new knowledge because we are now getting the after combining the results, we are getting a new answer or a new estimate. So there are four steps which we usually use while conducting the meta-analysis. First, we have to identify our study. We have to identify the data source from where we are collecting the data. We have to determine the eligibility because, and this is very important, the study selection, because there are hundreds of studies available. And out of these hundreds, suppose we have to analyze only 5 or 10. So how you are going to study, how you are going to select a suitable study is very important while conducting the meta-analysis. So we have to define the inclusion and exclusion criteria, which is which ones to keep and which ones to remove or throw out. So then the third step is collecting the data. To abstract data from the studies, we have to identify and collect the data. And finally, the last step is statistical analysis, or we have to do analysis of the data from the studies. So these are the four basic steps which we use while conducting the meta-analysis. Now, the next thing is how we are going to look for the meta-analysis. So one very good way to identify the systematic reviews and meta-analysis is by using a graph, and this graph is called as forest plots. What is this forest plot? The results of a meta-analysis are usually presented in a figure or graph, which is called a forest plot. The forest plot in the next slide, which you will see, is from a systematic review of a RCT, which assessed the effect of giving corticosteroid to women who were about to give birth prematurely. So this is the research question. As you can see, we are going to look at the forest plot of a research question, and the research question is the effect of giving corticosteroid to women who were about to give birth prematurely. So this is the research question, and a randomized clinical trials were done for this research question. And this is the next slide. Now you will see is the forest plot. Yeah. So this graph or this pattern which you are seeing some lines, some arrows, some squares, some diamond shape bar. So this type of graphical presentation or this type of figure, which you can say, if anybody asks you that what is the name of this graph or figure, so this is a forest plot. So now, why it is called as a forest plot? Why does it look like a forest? Not really. So just it was called as forest because some authors they because of these lines. These lines you can imagine that suppose this is the ground and these are lines. So these lines look like trees. So depending, suppose there are hundreds of studies there, so it will be a bushy appearance. So because of this bush-like appearance of these lines, it was called by forest as a forest-like appearance. It was called by forest plot by some of the scientists, and since then this name was adopted. So what points to look for in this? I will just explain to you. So as I told you, the research question, what was the research question for the systematic review? As you can see, it is written here. The review, the research question is mentioned, effect of corticosteroid before preterm delivery. So we are trying, this is a randomized, this is a meta-analysis. Okay, so this is a meta-analysis because for we are doing forest plot only for the meta-analysis. So Uh, yes, so as you can see here, what we are doing, we are trying to look for the effect of corticosteroid before preterm delivery. So there are two groups. There is a comparison group in which we are doing, we are looking for the effect between corticosteroids and placebo, and we are looking for the outcome like the neonatal death. So now on the left hand side, you can see that there are many studies mentioned. Okay, and you can see Liggins and Hobby 1972. So what does it mean? It means that all these researchers, all these authors have done the randomized clinical trial on the same topic. The topic is same, like the effect of corticosteroid before. So all these authors have studied the effect of corticosteroid before preterm delivery. And when they did this study, this is the year. Like Liggins, he studied in the year 1972. Then Block et al. Studied in 1977. Then the recently the collaborative in 1981. So all these researchers have studied this particular research question. The same question has been studied and the year mentioned. So usually in the forest plot, we arrange in the chronological order. As you can see, this is the oldest, and this is the recent study, 72, 77, 78, 79. So as we go down, the year increases. So we have arranged all the authors on the left hand side. Then on the right side, you will see that this is the odds ratio. We have calculated the odds ratio, and for the best possible results, we calculate for the 95% confidence interval. This is CI is the confidence interval. I will in the next few slides you will understand what is the meaning of this confidence interval. Then we give this weight percentage means how much weight or how much emphasis we give to the studies. Like you can see the study number one, and these bars, these horizontal bars, these are the confidence interval for the study. Like from here in the Liggins, from this, I think you can see the pointer from this side to this side. This is the confidence interval for Liggins. From this side of the arrow to the further this side is the confidence interval for the study from Mr. Block. So we have got the weight, then we have got the odds ratio, and this is the range of confidence interval. Confidence interval range, like in this case, it is mentioned 0.38 to 0.89. So you can see here also, as a just focus on Liggins and Hobby, the confidence interval is from 0.38 to 0.89. As you can see, I told you this is the confidence interval. Now go down. This is the bar on which you can see 0.39 is somewhere. I think for here it's right. 0.39 falls here on this point, and 0.89 just below one, because this line, yeah. So this line, this vertical line is called as the line of no effect. Line of no effect, it is usually 1, or sometimes in some graphs it may be 0 also. So on the left hand side, it is in less than 1, that is favors corticosteroid, and on the right hand side, from 1 onwards, it is that the studies are favoring placebo. So what we can see, like in this case, the study is favoring the corticosteroid. As you can see, the confidence interval is on the left hand side of the line of no effect. This is the line of no effect. So on the left hand side. Okay. And one more trick I can tell you to identify that whether the study is statistically significant or not significant. So I think I told you maybe in some of the classes before, we look for the significance level by p-value. As you can see here down, this is the p-value. P is equal to 0.0006. So if the p-value is less than 0.05, then the study is called as statistically significant. And if the p-value is more than 0.05, then the study is not significant, right? So just remember, this is an important point by which easily you can comment if anybody asks you that whether a study is significant or not. If the confidence interval is not crossing the line of no effect, as I told you, this vertical line is called as the line of no effect. If it is not crossing the line of no effect, then it is statistically significant. Okay, so like the first study, Liggins and Hobby, it is crossing the line of no effect or not? Yes, please answer. Anyone? Look
No.
at first. No. Yes. So it is not crossing the line of no effect. So this study is statistically significant. Look at the second one, Block et al. It is significant or not?
Not significant. It
Yes.
crossed the line. No
Yeah,
effect.
good. Because it has crossed the line, so it is not significant. The third study,
Also not significant.
not significant. Fourth,
Not significant.
fifth,
Significant.
yes, good. Next,
And the last two are not significant.
very good. So very easily by looking at the studies, we can comment by simply seeing that whether the confidence interval is crossing the line of no effect or not. Then the most important point, as I told you that in the meta-analysis, we are combining the results and we are getting a common result. So if you see the lower down, there's a diamond shaped figure. So this diamond shaped figure is the overall result or the overall effect, right? And the edges, the points of this diamond, this is the confidence interval of the overall analysis. So we are analyzing the results of all these different authors and we are getting a diamond shaped curve. Whereas in the individual studies, we are getting small squares. As you can see, square 1, 2, 3, 4, 4. Then also you must be noticing the different size of a square. Can anybody tell me what is the importance of this? Is it a small size? Is it a bigger size? Any idea? The bigger the size of the square, the, the bigger the size of square, the more is the effect or the more better is the study, or you can say more is the sample size. Okay, the more the sample size, the more is the weight or precision of the study, and more is the sample size. And also one more thing at this point I want to tell you that more the sample size, smaller the confidence interval. As you can see, the confidence interval is smaller and the weight is more and the sample size is more, right? So what do we want? Always we want a smaller confidence interval, because a smaller confidence interval means a larger sample size. The more the sample size, the better is the study, the better are the results. So just comment on the final result of this study. This diamond shaped figure, it shows the final result. It is significant or not?
Significant.
Significant, yes, very good. So also you can see in the values down, you can see that the test for this is called as the overall effect. This is called as the overall effect. You can see the p-value. The p-value is highly significant. I told you if the p-value is less than 0.05, then it is statistically significant. So it is, in this case, it is very less. It is 0.0006. So the results are statistically significant. So what will be your comment? That yes, the effect of corticosteroid before preterm, so it is favoring, as you can see, it is on the left side. This figure is on the left hand side of the line of no effect. So it favors corticosteroid. So it says that yes, there is a positive role, there is a significant role of corticosteroid before preterm delivery in the prevention of neonatal death. Whereas the placebos, they don't have any effect on the preterm delivery. So this is how we are doing the interpretation of the forest plot. Okay. So basically in the forest plot, you have to look for the graph, you have to look for these graphs, and the same thing is also mentioned on the right hand side in the form of the figures. I will explain to you, it will come to you. As you can see in this case, in the overall effect, as you can see, it is from the range of confidence interval is from 0.46 to 0.81. Just go down, it starts somewhere from here, 0.46 to 0.81. So it is less than 1. It is not crossing the line of no effect, so it is statistically significant. I think it should be clear to you. Yeah, so again, the same thing has been explained to you here in this particular forest plot. So it shows the uh, different types of studies. It shows the control group, the treatment or the intervention group. There is a p-value mentioned, which indicates the level of significance. If the p-value is like in this case, p-value is 0.7. So what is 0.7? It is significant or not?
Not significant.
Not
Significant.
significant. Yes, good. Like as you can see, the overall effect. This is significant or not?
Not significant.
Not significant because it is crossing the line of no effect. As you can see, the figure is crossing the line of no effect. So it is not statistically significant. This is the line of no effect, which is mentioned here. And these are the different types of studies. So as you can see, all the studies are not significant. All the studies, the study number A, study B, study C, all the studies are crossing the line of no effect. So they are not statistically significant. Okay. And the size of the squares, it shows the effect size of the weight. Like as you can see, the weight percentage is more in the study B compared to study A, and the weight percentage is lowest. It is just 4.5 in the. So the weight percentage is reflected in the size of the squares. So for individual studies, remember we use the squares, and for the common result we use the diamond. This diamond shaped figure is used to to mention the main outcome or the main result. So you can see again, each study is represented. These are different types of studies. This is the weight effect, the line of no effect. On the left hand side is favors experimental group, and the right favors control group. And these lines, these horizontal lines are called as the confidence interval. And the diamond, it shows the overall effect estimate. As I told you before also, it shows the overall effect estimate. As you can see, in this case, it is just crossing the line of no effect, just crossing. It is the range is from 0.36 to 1.15, from 0.36 to 1.15. So slightly it is crossing. So, so same you can see in the p-value. The p-value is not significant. It is 0.13. For significant, it should be less than 0.05. So very easily, just by looking at the graph, you can identify. So a forest plot is usually accompanied by a table listing the author's name and date. As you can see on the left hand side, we have the name of the authors and the date of conduction of the study. Then the table also lists the mean score, the standard deviation, the number of participants also you can see. N means the number of study participants. Okay, so all the information is there in the forest plot. So just to revise the interpretation of forest plot, the names on the left, these are the first authors of the primary studies. So these are the first names, the surnames of the different authors. And also it is, I think it's not mentioned in this table, but usually we write the year also in which year the study was mentioned. The black squares, as I told you, the black squares, these are the relative risk or the odds ratio of individual studies. So for individual studies, we are using the squares, we are using the black squares. And the size of the black square, it tells us about the weight of the each trial, weight or the you can see how much effect that particular study has. So the weight of the study is given by the size of the black square. Then the horizontal lines, these are the confidence intervals. These are the confidence intervals. Usually we use the 95% confidence intervals. Then we also have a vertical line, and this vertical line is the line of no effect. Line of no effect. Why it is called the line of no effect? Because in this case, the odds ratio or the relative risk is 1. So therefore, we say that it is the line of no effect. The diamond shaped curve, shaped figure, it shows the overall treatment effect. The diamond center, it shows the combined treatment effect. The tip of the diamond, these tips from on the left and the right side, it shows again the 95% confidence interval. So this is about the interpretation of the forest plot. Okay, then we the statistics which is relevant to meta-analysis. in the sample and population. So in a study sample, the samples are usually collected from a much larger group, which is called a population. A sample, why we collect a sample? Because a sample allows us to make the inferences about the entire population, because it's not possible for us to study the entire population. So definitely we need a small part of the population, which is called as a sample. So how we can quantify this sample? This sample can be quantified by using the confidence intervals. So now you will come to know that what is the basic meaning of this confidence interval. So basically, a confidence interval, it indicates how precisely or how effectively a sample mean estimates the true mean of the population from which the study sample was drawn. So suppose we calculate the mean, then the confidence interval is usually a range. Is usually a range of values which within which the true mean of the population is expected to lie. So it's basically a mean value, and it will tell you the entire range. And within this confidence interval, all the values of the true mean is expected to lie. So this is the confidence interval, and usually it is expressed as a 95% value, because it is expressed as a range of values around a mean. That is, as a lower, every confidence interval has a lower value and a upper value, as we saw in the forest plot, and it is usually chosen as 95%. So because at 95% confidence interval means that we will be what does it mean? It means that we can be 95% confident that the true population mean it lies somewhere between the limits of the interval. So we will say that all our sample size is within the limits of the interval, and we are 95% sure. So therefore it is called as a 95% confidence interval. In another way, if the study was repeated numerous times or multiple times, then 95% of the samples would produce the confidence interval that include the true population mean, and 5% would not. So always in most of the clinical trials, in most of the meta-analysis, we take the 95 because 5% is considered as error. We can't take it as 100%. It's practically not possible. So we take the 95% confidence interval in the studies. And there are different factors which can affect the size of the confidence interval. The most important is the sample size. As I told you that if the sample size is more, the confidence interval will be smaller. And if the sample size is small, the confidence interval will be more. So both are inversely related. So just remember this, that the larger samples will produce the narrower confidence interval or the smaller confidence interval. Then sample variability. Sample variability like the standard deviation or dispersion of the sample again will cause narrow confidence interval, because always it is good. Which type of, which is better, a narrow confidence interval or a wider confidence interval? So always narrower confidence interval is better, because once it is narrow, then it will mean that the sample size is more, and always when the sample size is more, there are less chances of error and the results are better. The results are always good. Therefore, we always try to take a bigger sample to reduce the confidence interval. Then the level of confidence chosen, a larger confidence level results in a broader. So don't confuse the confidence interval with the confidence level. A larger confidence level, it results in a broader confidence interval. The more certain we want to be that the interval includes the population mean, the wider the confidence interval will be. So don't confuse with this. Just remember the first part is very important, that simply by looking at the confidence interval, we can tell that whether the sample size is big or small. So for larger samples, the confidence interval will be narrower. Now, in the meta-analysis, it is good, but again, there are some bias which is involved in the meta-analysis. And the most important bias in the meta-analysis is the publication bias. So what is this publication bias? So in this, the selective publication of articles that show positive treatment of effects and statistical significance. So uh, what is being done in the publication bias? Like suppose I have done a research and now I have done a meta-analysis and now I want to publish it. So as a researcher, what do you think that only those results which are positive or which are statistically significant, they should be published. We try to we avoid publishing the negative results or the negative findings or those findings which are not statistically significant. This is the mentality of a researcher. He tries to publish only those results which are positive or which are statistically significant. So this is called as a bias because this is not correct, and this will lead to publication bias because we are doing the selective publication of only those articles which are showing the positive effects and which are statistically significant. Then another bias which is common in meta-analysis is called as the English language or the duplication bias. So usually in this case, the studies with significant results are more likely to be published in English. So they think that in the English language, if the results are significant, then only they will be published. So this is just a thought. That's a thought of a researcher. This is a bias. Then also there is a citation bias, and it occurs when the studies with significant or positive results they are referred or referenced in other publications compared with studies with negative findings. So again, people think that only those articles will be referenced or cited if the results are positive and statistically significant. And they think that if the findings are negative, then usually it will not be cited in the citations. So this is again a type of bias. Yeah, so same thing that publication bias, it implies that those studies which show statistically significant results have a greater tendency to be published. So this is the thought of a researcher. He is always thinking that if the study is positive, if the results are significant, only then they will be published and the negative findings will not be published. So this is the bias which occurs in the meta-analysis. The publication bias is the most common type of bias which usually occurs in the meta-analysis. So how we will detect it? How we can detect this publication bias? So we can detect it by using a method or a technique or a plot, and this is called as a funnel plot. So we use the funnel plots to detect the publication bias. So as you can see in this figure, uh, like on the left hand side, you can see there are two types of drug effects. In this case, this is the positive finding, the treatment works. And on the right hand side, you can see the treatment does not work. So this is a good study. It shows a symmetrical normal distribution that uh, uh, it is showing the results of both and the treatment positive results and the treatment negative results. Whereas in this type of study, you can see this is the asymmetrical result in which only the positive results are mentioned. The treatment works, whereas there is hardly any study which shows that the treatment does not work. Okay, so this is the asymmetrical study. So it shows a gaping hole where the negative study should be mentioned. So this is asymmetrical, and as you can see, this is funnel-like. As like in the you can see this is just like a funnel. So therefore, it is called as a funnel plot. So this asymmetrical appearance of the funnel plot. So if this funnel is asymmetrical, then we will say that yes, it is asymmetrical because of the publication bias, because the negative studies are not published by the researchers. So this asymmetrical appearance of the funnel plot with a gap, there is a gap now present, as you can see in a bottom corner of the graph. So this shows the publication bias of the results. Now the outcome variables in meta-analysis can be either the binary data, which can be there is only two data or two outcomes like yes or no or positive or negative, or it can be a continuous type of data. So binary data, for example, an event rate like something that either happens or does not happen. That is a treatment or failure. Healthy, like there are two sides of a coin. So this type of data is called as a binary type of data. So this binary data is usually presented in the form of odds ratio or risk ratio. Okay. So the data which is presented this binary data is presented using the odds and risk ratio. Whereas, yeah, so this is the interpretation of the odds and risk ratio. If the odds or risk ratio is more than 1, it shows that the outcome occurred more in the intervention group. If it is less than 1, it means it occurs less frequently in the intervention or the treatment group. If it is 1, it shows that there is no difference between the two groups. So by looking at the values of the odds ratio and relative risk, we can comment that whether the study is favoring the treatment group or whether the study is favoring the control group. As you can see, again, this is a forest plot of a meta-analysis. You can see that the overall effect or the diamond is on the left hand side. It is favoring the treatment group. Okay, on the left hand side is the treatment group. On the right hand side of the graph is the control group. And also it is that, as you can see, the tip is slightly crossing the lines, and so the p-value is exactly not less than 0.05. It is 0.09. Okay, so we can just also comment on whether it is significant or not. So the continuous data will have the differences in their mean value. Okay, so I think no need to go into further details, the weighted and the standardized mean differences. Just we can focus on the forest plot and look on the, like, suppose like these are the squares for the individual studies. So as I told you, the larger the size of the square, the more will be the effect size. So these are for the individual studies. All these studies, study number 1, 2, 3, all are not significant because they are crossing the line of no effect. And also the overall effect is also not significant because the tip is slightly crossing the line. Just if you will enlarge and zoom it, it is slightly crossing the line. Also, we can check with the values of the confidence interval. So in a meta-analysis, when there is no difference between the groups, the mean difference is 0. The value of the line of no effect is 0. A negative value means that the outcome is favoring the treatment. As I told you in the graph, as you can see, if it is on the left hand side or the negative side, it is favoring the treatment. If it is on the right hand side, it is favoring the control. A positive value means that the outcome favors control, indicating that the treatment is harmful compared to no treatment. Again, the same, this is the line of no effect. So because the overall effect is on the left hand side, it is favoring the treatment group or the experimental group. So where to find the systematic reviews? You can find them in a variety of journals, the good published or indexed journals. If you are searching on electronic database, you can use the Medline. You can narrow down your search down to systematic reviews under the types of publication. In addition, if you really want to study about the systematic reviews, you can search the Cochrane database of systematic review, and this is the website for Cochrane collaboration. So there is a Cochrane collaboration, and under this collaboration, there are thousands of systematic reviews for different types of studies available. So if you will log in to it, then you can find out the reviews for different types of studies. So depending on your interest type, you can search those and study those systematic reviews. So from where the name of Cochrane has come? So this name Cochrane, it was named after Mr. Archie Cochrane. So Mr. Archie Cochrane was a famous British medical researcher and who mentioned the urgent need for systematic reviews of high quality research. So under his name, this Cochrane collaboration was developed. So this is the, as you can see, this is the photograph of Mr. Cochrane, Professor Archibald Cochrane, and this is the Cochrane collaboration. So, and this is just if you can see the symbol of Cochrane, the symbol of Cochrane, it is the forest plot. You can see in the symbol, there are 7 studies, 1, 2, 3, 4, 5, 6, 7, and this is the overall, the diamond shaped curve. So this is the symbol of the Cochrane collaboration. So systematic reviews, as I told you in systematic review, we have to make a protocol, we have to prepare a protocol, a research question, we have to review it, and ultimately, and we can look for the similar type of studies in the Cochrane library. So depending on the type of the study, we can use it. We can, and these protocols are frequently updated and revised, and the standard systematic review protocol should include the study objectives. It should include all the criterias for the review, the search strategies for identifying the studies, how you are doing the study selection, the inclusion criteria, exclusion criteria, the data analysis. So they are considered the best source of the best type of evidence, and they form the apex or the top of the EBM pyramid. So this is the Cochrane collaboration logo. As I told you, it looks like a forest plot of 7 randomized clinical trials. Thank you very much. So this was a brief session about the systematic review and meta-analysis, and you will practice it more in your practical sessions. Then even if there is some doubt, I think it will be uh, made to you clear or explained to you in the practical session. You will solve some of the examples and you will be told how you are going to interpret the forest plot and do the analysis. So if there is anything more which you want to know, kind of question, any questions?
Thank you.
Okay, thank you so much. So with this, I end the session. And inshallah, if you have any doubts, anything, it can be, you can make it clear in the practical session. Thank you so much. Assalamu alaikum.